Okay, welcome everyone to the IMAG Interagency Modeling and Analysis Group Multiscale Modeling webinar. This is a new grantee webinar, and Dr. Ronan Fleming is a member of a new grant directed by Michael Saunders, and the U01 award is titled Multiscale Molecular Systems Biology, Reconstruction and Model Optimization. And Ronan is presenting to us from the University of Luxembourg, where he is currently a senior research associate, and previously he was the independent group leader from the Center for Systems Biology at the University of Iceland, and we are very pleased to have Ronan present for us today. So welcome, Ronan, and thank you.
Okay, welcome live and accessory viewers. So I'll just get started. This is a presentation of a U01 award that's recently started, and it involves a number of partners in a collaboration. Inez Thiele in the Molecular Systems Physiology group at the University of Luxembourg, Professor Bernard Paulson and Systems Biology Research Group in San Diego, Professor Michael Saunders and the Systems Optimization Laboratory in Stanford, and myself also in Luxembourg. So what's in the title? I'll go through this slowly because different words mean different things to different people, but essentially by multi-scale we mean in a very general sense, in the sense that any model that would involve variables spread over many orders of magnitude. To us, that defines a multi-scale model. Molecular in the sense that the chemical formula at least is known for each molecule, so there are no arbitrary nodes in those networks. And system in the sense that it's a system of biochemical reactions with defined boundary conditions, and biology in the general sense, because this modeling work has biomedical, biotechnological, and environmental applications. So it's not just applications in medicine, it has applications in bioenergy and remediation, etc. Reconstruction is an abstraction of select biochemical, genetic, and genomic experimental data about a chosen biochemical system. And that's distinct from a model, which is in general it's a mathematical model, but combined with a particular reconstruction, one can then form a particular computational model for a particular reconstruction. So the mathematical model might be the same across species, but the actual computational model that would result would be very different depending on what input reconstruction one will use to particularize the model with. An optimization in the sense of numerical optimization, so with a firm grounding in mathematical optimization theory, because there are branches of optimization that use exact optimization, and this is distinct from that, and also branches of optimization that aren't very well grounded in mathematical optimization theory. So the background to this is that the genome scale computational models are increasing in size, and so progressing from single microbe to whole microbial communities, for instance. So a single microbe might have 1,000 reactions in metabolism, but a community of 1,000 species would then have 1,000,000 different reactions. So that's an example how one can just get an increasing size from having multiple microbes together. Increasing ratio of fastest to slowest timescales. So for instance, in a metabolic model versus a model with metabolism and macromolecular synthesis, the metabolic reactions are occurring about 10 to the 6 times slower or faster than the macromolecular synthesis reactions. So if one is a network where those two parts are integrated, then automatically you have 10 to the 6 different or 6 different orders of magnitude difference in the timescales of the variables. And increased comprehensiveness in terms of simulation fidelity. So how closely the model is representative of physical, chemical, and biochemical knowledge. So there's a trade-off here between making models that are, let's say, partially representative of reality and computationally scalable, like let's say linear approximations, versus highly high fidelity models that might have a lot of nonlinearity and as a result impossible, perhaps impossible to solve on a large scale. So there's a trade-off there to be made. And these three things lead to a mathematical and numerical optimization challenge, in the sense that one leads to large-scale numerical optimization problems. And the challenge there is to reduce the computational complexity of the algorithms to solve those problems. It's also a multi-scale numerical optimization. Most optimization software is ideal for variables of order 1, and also it's a mathematical formulation challenge, because as I mentioned, biochemical function, if you want to have a high fidelity, one has to represent inherently nonlinear processes, and the challenge there is how to formulate the mathematical representation or abstraction of reality in a manner such that it's amenable to solution with algorithms that have low time complexity. So ideally here we're looking for polynomial time algorithms. So while one might, let's say, achieve a quick buck or a quick improvement by going for algorithms that of high computational complexity that can represent or solve problems that are of intermediate difficulty, ultimately, because the models are going to get so large, to me, it doesn't make any sense to waste time on anything other than algorithms that are really scalable, even if they take longer to develop. So the history of this collaboration, it goes back to sometime in about 2008, when the Inez, Tila, and I were both in Professor Paulson's lab in UCSD. At the time, I was working on methods to incorporate extra constraints into constraint-based models, and Inez was working on generating the first reconstruction of macromolecular synthesis. So what happened at the time was that Inez had a new model which was generated from an integration of metabolism and macromolecular synthesis, which I will detail later. And this model was the first to have multiple scales, in the sense that metabolism and macromolecular synthesis were there. So you had these 6 orders of magnitude in different timescales. And also it was a very large model, so that was gone from typical metabolic models that might have 2,000 or 3,000 metabolites and 3,000 or 4,000 reactions to 40, 50, 60,000 metabolites or molecules. Let's say, in the general sense, 60,000 molecules, so maybe 80,000 reactions, and then 6 orders of magnitude difference between the timescales. So we realized then that there was an issue here, that we needed to bring in the expertise in large-scale numerical optimization to help us deal with this problem. And it was then that I approached Professor Saunders, and we set her to try to collaborate. And then collaboration grew into 2009. We were awarded a US Department of Energy grant from 2009 to 2012, where we worked on the early stages of this work, which then formed the basis for the new award from the Multiscale Modeling Consortium. So there's quite a long history there, let's say, and it's at the stage now where we're delighted to have 5 years more funding, because we really have built up some good momentum here. So just to go back to introduce the basics here. Reconstruction of reaction stoichiometry is illustrated in this figure here, where at the if you can see the little pointer here, these are a set of reactions. So this reaction triose phosphate isomerase will be represented by the consumption of one molecule of DHAP, which is here, and production of one molecule of G3P, which is here. So each reaction corresponds to one column of this stoichiometric matrix, and each molecule corresponds to one row of that matrix. So in this case, they're metabolites and metabolic reactions, but the exact same thing can be done for any biochemical reaction. So this is, I illustrated with metabolism, as I say, but it's just as applicable for metabolism as any other aspect of biochemistry. And in this case, Inez reconstructed the macromolecular synthesis machinery for E. coli during her thesis, and I was also involved in that work. And it tries to put in the mechanism behind the transcription and translation of every gene involved in the process itself. So it's this cyclic dependency within biology where one needs to have enzymes or proteins to do functions, but some of those functions might involve actually making enzymes or proteins. So in this sense, that cyclic dependency is already evident in this reconstruction of macromolecular synthesis machinery. Now, that reconstruction was of a single scale because it was macromolecular synthesis, whereas at a later date then it was integrated with metabolism. So the integration of metabolism with macromolecular synthesis, one can see the dependencies here. So from metabolism you get nucleotide triphosphates, etc., cofactors that are required by to create macromolecules, and those macromolecules in turn are used to catalyze the reactions of metabolism. And the whole model then, it requires, of course, from the boundary, carbon, nitrogen, sulfur, phosphorus sources, etc., and byproducts. So it has a defined metabolic boundary, but internally there's this dependency where metabolism produces the building blocks for macromolecular synthesis, and macromolecular synthesis then catalyzes the reactions in metabolism. And there's the main feature of this in terms of multiscale is this difference between the steady state reaction rates that one might expect in metabolism in a millimole per gram dry weight per hour, let's say if it was for monitoring bacterial metabolism, versus nanomolar per gram dry weight per hour for macromolecular fluxes. Although this is an example from E. coli, the same phenomenon will be present in any cell. So let's say if we take it just from there and say that is the reconstruction. When some people say, let's say, I'm not a fan of the constraint-based reconstruction analysis approach, well, everybody to build any kind of model would have to reconstruct the network themselves. So one can disassociate those methods from the following actual modeling method that might be used to bring the reconstruction alive, so to speak. So to bring that integrated reconstruction alive, various things had to be done. So, and two examples here is in metabolic reactions, there's an implicit representation of enzyme reactions in metabolic reconstructions, but in order to integrate metabolism and macromolecular synthesis, it was necessary to explicitly represent enzymes in the reaction. Furthermore, in a steady state model, without any other constraints, one can see here from this reaction that the enzyme is recycled from beginning and to the end. So at a steady state, there might be no necessity in the model to produce any enzyme. However, and one can enforce the necessity to produce enzyme by coupling the rate of utilization of an enzyme with its synthesis. So that's this coupling constraints here, which is coupling two different timescales together. So one timescale would be the use or the catalysis reaction involving an enzyme, and the other timescale would be the synthesis of that particular enzyme. So this is key really to enforcing this cyclic dependency between different reaction rates in the model. The different dependencies are between different parts of an integrated metabolic and macromolecular synthesis computational model are illustrated here. So DNA is represented at the sequence specific level. So every base that codes for that's protein coding would be represented, and the DNA of course gives rise to the mRNA, tRNA, and ribosomal RNA, and then those together go to form proteins and ribosomes, which are which also have their dependencies, and then, as I mentioned before, metabolism has this dependency on the proteins receptor. This is all biology. However, it's hard-coded inside the reconstruction, and the model represents this. So once it's reconstructed and implemented in the model, it's like having a perfect memory. The model will never forget. So, the main difficulty then from a multi-scale modeling perspective is that we would, for the optimization problems involving the large stoichiometric matrices illustrated here, you might have 60,000 rows and 80,000 columns, 10 to the 4 orders of magnitude difference in size of the stoichiometric coefficients. That's the magnitude of the entries in each part of the matrix. And this corresponds to the issue where many metabolic moieties are required to create one macromolecule, and also the reaction rates vary over many orders of magnitude. So this is not an artifact of the way in which the reconstruction is done. The reaction rates do vary over many orders of magnitude. Of course, one could split up a reaction where many metabolic moieties go to form one macromolecule into many smaller reactions that just add one moiety at a time, but then you would have many thousands more reactions and many thousands more molecule species. So it's a trade-off. So we presented this problem involving multi-scale flux balance analysis as one of the core things to be solved in the first grant that the DOE funded us for. And a succession of Stanford students approached this, and thankfully Yukai Sun in Michael Sonner's group was able to actually solve it. And it required digging into the actual matrix, because if one just took it and didn't try to learn, let's say, from the biological examples that were there, it wouldn't have been so easy to solve this problem. But the essential issue was to reformulate the problem and lift it so there were increased number of rows and columns in the matrix. But this is done in a way that gives you a trade-off between computational efficiency and reliability. So having it very compact and multi-scale leads to an unreliable optimization problem, in the sense that one could not tell the difference between an optimization problem that had got a solution and an optimization problem that didn't have any solution at all. So that was the unreliable aspect of it. But this could be, Yukai found a way to remove this unreliability at the cost of making the problem a slight bit slower to solve. But we prefer to be able to solve the problem and know that it's solved rather than to a certain accuracy, rather than to wonder whether it's been solved at all. So here you can see here in this table of results that before and after lifting, which is this procedure to reformulate the problem, the simplex solver requires more iterations to solve, but the worst in feasibility, that is the degree to which the problem is solved, increases. So here in the barrier case, a flux, a steady state might be violated to the 10 to the negative 1 in before this procedure, and then afterwards it's solved to 10 to the negative 6, which is acceptable. So this is especially important where the values of the fluxes might also already be small. So if the macromolecular synthesis fluxes are like 5 or 10, as in the first decade, then if one might not be able to tell if 0.1, let's say, is noise or whether it's a real result, but 0.0001 is much smaller, and it's clearer to see that there's a difference between the two. So this might be something what people have overlooked in the past. This is especially with these models is to be able to detect the difference between something that's small because it's real and something that's small because it's an artifact, and that's essentially the difficulty that you can overcome. So what's the effect of these integrating these two levels of reconstruction together in a model? Well, one can conduct what's called flux variability analysis. So that's computing the maximum and minimum rates through every reaction in the model. And what one can see here that the fluxes of the maximum minimum fluxes in a reaction in the integrated models, the me matrix, as metabolic and expression matrix as it's called, that difference between the maximum and minimum is much smaller than the same for the same reaction in a just a metabolic only. So this is indicating that the solution space, or the set of steady state fluxes, is much smaller in the integrated model than it is in the metabolic model alone. So that's from, let's say, a more numerical perspective. From a biological perspective, we can see already some insights that have been gained from using these multi-scale models. So this first example I'll give is of the mechanics of the genotype-phenotype relationship. So just to introduce this, we'll just revise the genetic code. And the genetic code has redundancy but no ambiguity, in the sense that there can be multiple codons per amino acid indicated here in green. There can be multiple tRNA molecules that read the same codon indicated in blue. And there can be, each tRNA can read multiple synonymous codons and may be able to read multiple synonymous codons indicated in yellow. So in this context, codon usage bias is the frequency of synonymous codons, and it differs between organisms within genomes and the long genes. So the question is, why does an organism or a gene or a genome have a particular codon usage bias? And there's a voluminous literature on this topic, but it has never to date been modeled mechanistically, as in that all the molecules, etc., would be actually represented in a computational model. It's been modeled phenomenologically, where there's some mapping and between a codon usage, let's say, and some phenotypic characteristic, but not all the mechanisms that might go from codons all the way to, let's say, growth rate. E. coli, it has its genetic code is illustrated here, and it has 86 tRNA molecules, and the number of, in parentheses here, is the number of distinct tRNA molecules per amino acid. So we mentioned before that multiple tRNA molecules can recognize a codon, and multiple codons can code for the same amino acid. So then you can have multiple tRNA molecules for each amino acid. For instance, leucine here, it has 6 different synonymous codons and 8 different tRNA molecules that recognize those codons. And in the wild type E. coli, CUG is the dominant synonymous codon. In other words, it's the one that's most there's the most bias towards that codon for leucine. In this integrated model of multiscale model of metabolism and macromolecular synthesis in E. coli, what was done is to take the wild type genome and either equilibrate the codon bias in other words, make it less biased than it was previously, or to make it more biased or biased in a different way than the wild type mean matrix. So the algorithm for generating the biased model is illustrated here. So you'd randomly choose a codon, identify all possible synonymous codons, and then choose randomly one synonymous codon and replace all of the first codon with the other synonymous codon. So then one would have to update the reconstruction based on the new genome sequence for E. coli, let's say this E. coli strain, this biased strain. And as a result, then one would have to change the transcription reactions to mRNA degradation reactions and translation reactions, etc. So it actually generates a new instance of a mutant E. coli in silico, let's say, a biased mutant E. coli, and likewise for the equilibrated strains. So that's what's pictorially represented here. And then what was done was to compare the function of the biased strains with the wild type strains with the equilibrated codon usage strains. And what was found is that the changes in codon usage affect the ability to grow. So for instance, some it's not illustrated here, but for some growth media, so 170 different growth media were tried in silico, and for some bias strains, they were unable to grow where the wild type was able to grow on particular media, and for other media, the wild type was not able to grow, but the bias strain was able to grow, and and also then shown in this figure is that the maximum possible growth rate in silico for the E. coli integrated multiscale model was different relative to wild type. So in these two figures here, B1 to B9 are B10 B1 to B10 are the biased strains, and EQ1 to 5 are the equilibrated strains. So the growth rate shown here then is relative to wild type. So some bias strains, as B2, B5, and B9, they had the same growth rate relative to wild type on glucose aerobic medium, let's say, and also on lactate medium. But other bias strains had much slower growth, so down near 50% of the growth of the wild type. Now, this type of information would have been available experimentally in some cases. So that change in the codon usage bias might affect the maximum possible growth rate would have been something that would have been seen experimentally. However, the main point here is that one can now delve into the results from the simulation, as in analyze actually the flux vector, the steady state flux vector that arose from trying to optimize the growth rate in a particular condition, and look and see why is that growth rate not as high as it was previously in the wild type. And this is not possible to do experimentally in as efficient a manner as it is possible to do in silico. So the idea here is that it's possible to delve into the mechanisms relating the actual codon usage or sequence for an organism to some phenotypic characteristic like growth rate, and all the way in between in the molecular steps. And this can be done with the model. So what was done in this case was to look at the numerical properties of the flux balance analysis solutions, and what was able to find after some searching was that the limit to growth rate was ribosomal RNA operon transcription rate in the wild type, but in some of the biased strains it was the leucyl tRNA transcription rate that was limiting growth. And the technical thing that was searched for here is called the reduced cost, but that's for the aficionados of COBRA modeling. But suffice to say that if we just give this example here, let's say for leucine, the CUG codon can be recognized by 1, 2, 3, 4, 5 different leucine tRNA molecules. Now, if one takes that codon and in a biased strain switches it over to CUU, the CUU codon can only be recognized by 1 tRNA molecule. Now, because there's a limitation on the rate at which a tRNA molecule can be transcribed, there's a demand, the increased demand caused by the change in the dominant codon for leucine to CUU can't be met, because the supply is fixed by the transcription rate of the tRNA molecule. So in this case, for the first bias strain, the switch to CUU was reduced the growth rate, which in lactate and glucose aerobic medium, which you can see here. But if one switched to a dominant codon UUG that had 2 cognizant tRNA molecules, then one didn't see a growth reduction, and that's here in the B5. And so let's say if one has an increased tRNA demand, this can be met by augmenting supply. So that might either could involve modification of a tRNA to expand its set of red codons. And in fact, there's a strain of E. coli where the tRNA LUW is also able to read CUU. So that would give rise to a second codon here to read the CUU molecule in addition to LUU, and perhaps alleviate the growth rate limitation. So this is an indication that in fact in nature the expansion of the set of red codons by a tRNA does actually occur. So it remains to be experimentally established if E. coli can indeed or not read CUU, the LU tRNA. So I guess these details are highly mechanistic and of most interest to people who are into codon usage bias. But the idea here is that the models, because they're a molecular level, they can relate, and also that they are of a large scale. They can relate everything from a phenomenon that are detailed on the genetic level with phenomena that are detailed on the growth rate. So another example of this is iterative annotation of gene function, and this was this came out from the second multiscale model created from Termatoga maritima, and here there was comparison of the in vivo and in silico transcriptome for Termatoga maritima growing on L. arabinose or cellobinos, minimum medium, and the in silico expression of genes upregulated and downregulated was confirmed when, in fact, they went and measured the expression of these genes in the lab. And what was done then was to scan the promoter regions of the genes that were up and down regulated and identify high scoring motifs that correspond to sets of these up or down regulated genes. Then one could use that motif to search back across the rest of the genome and find genes that might be regulated in the same manner. So in fact, what they found was there was the AAR motif identified 3 genes, one of which was had a sequence similar to a sugar binding protein for arabinose ABC transporter, and 2 others which were permeases of the ABC transporter. So at the time, there was no annotation in the Termatoga genome of these 3 genes, and by this process, an iterative workflow through comparison of gene expression, experimental data, and bioinformatics was able to re-annotate some new genes. And then, of course, this could be fed back then into the model into the reconstruction to produce a better model, better predictions, new annotations, and so on in an iterative workflow. So those are 2 examples of the use of COBRA modeling in multiscale molecular models. At the current status, there's only 2 of those models, but here is a figure of the cumulative number of metabolic reconstructions that are available. So there's 139 unique models available, unique species available to date that have metabolic models, and we envisage that most of these species will generate their own multiscale molecular model in the near future. The reason being, as I've outlined previously, is that the set of steady state solutions amenable to these models is smaller, and one can investigate aspects that are hard to investigate in metabolic models alone. So that's the aim of the systems biology group in UCSD, is to develop algorithmic workflows and software to allow everyone to generate their own multi-scale models. So hopefully by 2017 we'll have tens of multi-scale models generated by other groups throughout the world. For the next 5 years, the aims in Stanford are more fundamental. So at present, if one looks at the history of scientific computing, the variables and the computation have been done in first in single precision, and now these days it's all double precision. And what Stanford will do in the coming years is to develop quad precision versions of all of their key linear programming, quadratic programming, and nonlinear programming solvers. So this, I think, will be of interest in general to everybody involved in multiscale modeling, not just in molecular systems biology, because these types of solvers are in general use in many simulations. So, and the idea here is that the number of significant digits that these solvers can represent increases. So they're intrinsically more adept for dealing with variables that span over many orders of magnitude. So also, as we mentioned, that the reconstruction sizes are increasing. So here you can see from 1990 to 2009 the rise in the number of reactions in these models. And the other aim at Stanford is to develop a hypergraph network flow algorithms for optimization with biological networks. So the background here is that standard network flow algorithms have a complexity of order n. In other words, the amount of time that in worst case it would take to solve it goes up in proportion to the number of variables. Whereas standard linear optimization algorithms go up with order n to the 3. So the issue here is that the network flow algorithms have been designed specifically for graphs, whereas biological networks are hypergraphs. And so that's forced us to use the standard linear optimization algorithms. But perhaps, and this is speculative and it's an open research question, that there might exist an algorithm of lower computational complexity than the standard linear optimization solver that would be specific for biochemical hypergraph network flow problems. So this is, let's say, a high risk, high gain aim, where if we can succeed in this, we will have a class of solvers specifically for biochemical network problems. Biochemical networks are also increasing in scope, so not just metabolism, macromolecular synthesis, but also transcriptional regulation, signaling, etc. And there's the issue here is that currently the only large-scale method for modeling these networks represents explicitly reaction flux, but not molecular abundance. So the abundance of the concentration of metabolites is not explicitly represented. So the question is, does there exist a scalable approach to model fluxes and concentrations explicitly in some manner? And that's what's mainly focused on by my group in the University of Luxembourg. So there's no way to describe this without some details. So if we just consider a biochemical network with n molecular species and n reversible reactions, we can define forward and reverse stoichiometric matrices for the forward and reverse reaction stoichiometry. And we assume that each reaction is mass balanced, and let C be the metabolite species concentration. And assuming elementary reaction kinetics, one gets rate laws of the form showed here. And then the deterministic dynamical equation for evolution of molecular species concentration is given here. So this is the same as most people would be familiar with, but perhaps in slightly different notation. Now the issue here is that at steady state, where there's no change in concentration over time, a non-equilibrium steady state where the forward and reverse reaction rates don't equal each other, one is interested in finding a set of concentrations that satisfy the steady state equation. Now the problem with that is that the set of steady state concentrations is non-convex, whereas the steady state flux is convex. Now there's a big difference between those two, and this is a non-convex set here, and this is a convex set here. It looks like a small difference, but if you combine that with high dimensionality, it becomes very difficult to solve these models in an efficient and reliable manner. So where one needs an algorithm with that are efficient, that are guaranteed to converge and provide a certificate of infeasibility, in the sense that the algorithm would come back and tell you, look, your model admits no steady state. It doesn't exist. There's no point in looking for one. The other issue there is that the kinetic parameters vary over many orders of magnitude. So for the molecular species concentrations, they vary over many orders of magnitude. So you can imagine the transcript of abundance versus metabolite abundance is much different. And the other problem there is also the paucity of kinetic parameters. So it's a lot of the kinetic parameters for enzymes, etc., are unknown. So we'd like to be able to use some of the optimization techniques that have worked so well in flux balance analysis, and yet also increase the scope into modeling concentrations. The upside here is that, in fact, there are ways to incorporate nonlinearity into these models if one uses convex optimization, and that's the essential core of the work we've been doing is to apply convex optimization methods to help incorporate extra biochemical fidelity and physical chemical fidelity into these models. So we published a paper where we described the first polynomial time algorithm, which is able to solve for steady state fluxes and chemical potentials. Now, the limitation there was that one was only able to constrain the ratio of concentrations, not absolute concentrations. And the second one was that biochemical reaction directions are an evolved set of those that are thermodynamically feasible. So we still have improvements to make. Furthermore, on the parameter paucity issue, we've been at the forefront of methods to develop new ways to estimate what the bounds are on the ratio of forward over reverse kinetic parameters, and this is described in detail in this recent paper here, where we devised a method that makes the best out of multiple available methods to best compromise between multiple available methods for estimating the bounds on these parameters. Furthermore, we've published results, first results actually to demonstrate easily testable conditions that will be, one is able to check to see if there exists at all a steady state concentration for a particular model. So that's the question of trying to see, does a steady state exist at all in the first place before trying to turn an algorithm on the problem to see if one can find it. And a surprising number of networks out there don't satisfy these conditions. So the main aims for the systems biochemistry and molecular systems physiology groups in Luxembourg are to develop scalable algorithms for multi-scale reconstruction and modeling. So this will be software to enable the reconstruction of biochemical networks in an accurate manner, and what we would like to call multi-scale mass conservative elementary kinetic modeling, which is the framework I touched on earlier, where the forward problem is to, if one was given kinetic parameters, to compute the corresponding non-equilibrium steady state. And then the much more challenging inverse problem of, if you're given reaction stoichiometry, experimental boundary conditions, thermodynamic constraints on net reaction directions consistent with biochemistry, to search amongst the possible kinetic parameters that might satisfy certain optimality criteria. So this is a way of trying to not fit parameters so much as to find parameters that satisfy certain optimality criteria, and to then check if these give rise to models that make predictions in agreement with reality. The way we'd like to approach this is via gradient-based search of kinetic parameters, and that differs a lot from existing approaches. There are many people trying to develop algorithms for fitting kinetic parameters, and most of them that I've seen use evolutionary computation or some method of optimization that tries to interpret the problem as something having local minima and try to solve it in a manner that tries to it's hard to explain, but basically if your problem has a local minima in it, it's difficult to solve it with any algorithm. And I believe that the solution to this problem involves in looking at it in a different manner and reformulating so it doesn't have local minima. But it's also a high risk and high gain project, because if we succeed, then we'll be able to do this in high dimensions, whereas the current algorithms are only able to do something up to about dimension 100, which makes them useless for the size of metabolic models or even the integrated multi-scale models that we have already for E. coli. So we also share all of the models that's been generated through this collaboration. Most of them are shared with the systems biology markup language in that format. However, for the multi-scale models, a standardized representation still has to be developed. The models can be downloaded from the links provided here, and also we do share all the software we developed quite quickly, actually, after the results have been published using them for the first time. So there's a reconstruction tool, or Bionet, a tool for thermodynamically constraining the kinetic parameters in a model, or assigning reaction directionality. von Bertalanffy, a very popular toolbox for quantitative prediction with constraint-based models. I noticed the other day that paper actually has been cited 130 times in the last 2.5 years and 90 in last year alone. Python software for doing the same, fast algorithms for doing flux variability analysis, for robust flux variability analysis, and also the routines for the basic engines of the solvers provided in Stanford are also open source. And we also support the community's efforts to use the COBRA modeling with this very active Open COBRA Google group, and anyone can log on there and see, well, join as a member and see that since 2009 there have been 430 topics. Actually, there's more than 430 posts. There's 430 topics have been discussed. There's 300 different members, and if you ask an intelligent question, you will definitely get an answer. We get a lot of people who will ask a question when the answer is simply to read the paper. However, if anybody asks a reasonably thought it through question, they'll get an answer within a day or two. So we provide software and models to the community, plus we provide support for people who want to try and use them. So that's it. I'd like to thank the colleagues in the Multiscale Systems Biology collaboration, all the students, etc., who I've cited via their papers on the previous slides. And I'd like to thank funding from the National Institute of Health and the US Department of Energy for the next 5 years for this project, and also for the coordination from the Interagency Modeling and Analysis Group, which I am just 1 year in so far now, and enjoying the broadening of perspective. Thank you.
Thank you very much, Ronan. That was a great presentation. Thank you for walking us through your award. And as Ronan mentioned, this is a co-funded cooperative agreement UL1 award between the Department of Energy and the National Institute of General Medical Sciences. So it's great to have this collaboration between the IMAG agencies as well. We'll open up for any questions. So would people please show their videos and turn on their mics, and we'll have an open discussion. Well, I'll start. Ronan, would you be willing to have some of these links that you've shown about your open source software posted on the IMAG wiki, so the people in the consortium will have more easy access to use it?
Sure, yeah. Yukai Sun is maintaining, at Stanford, is maintaining the website. I'm sure he'd be happy to help you with that.
Great, thanks. Any questions for Ronan? There's somebody's video that's showing gray. I'm not sure if
it can't all have been that clear.
Well, we will be posting Ronan's slides and this recording on the IMAG wiki, and I suppose we can include your email if people need to ask you questions after the fact. Okay, well, if we don't have any questions, I want to thank you again, Ronan, for giving us this great presentation. And we'll look forward to hopefully seeing you at the Multiscale Modeling Consortium meeting this fall.
Very good.
Thank you.
Okay,
Thank you, everyone.
Thank you. Thank you. Bye.